Welcome to Lecture 13, in which we will discuss biotechnology and how we can use biological systems to create goods and services that benefit society. This is Dr. Jennifer Doudna and Dr. Emmanuelle Charpentier, who both received the Nobel Prize in 2020 for the discovery of the CRISPR-Cas9 system. This discovery greatly progressed biological research, and in 2023, the first CRISPR-Cas9 based gene therapy was approved to treat sickle cell anemia. Our learning objectives for this video are to describe how restriction enzymes, PCR, and gel electrophoresis work, discuss the applications of biotechnology in DNA fingerprinting and molecular cloning, describe how CRISPR-Cas9 works, discuss the applications of biotechnology in health and agriculture, and discuss bioethics. Restriction enzymes were first discovered as prokaryotic defense mechanisms against foreign DNA, particularly viruses. They are also known as restriction endonucleases because they cut within the DNA rather than at the ends of DNA. They recognize and cut at specific palindromic DNA sequences, with two examples shown here. Palindromic sequences are DNA sequences that are read the same way regardless of which strand they're on, such as here where it is GAATTC from 5' to 3', whether it is on the top strand or the bottom complementary strand. When the enzyme makes a cut, it forms fragments. These fragments can have sticky ends, which are unpaired sequences that create overhangs at the ends of the DNA, or blunt ends, which don't have any unpaired sequences. In prokaryotes, the foreign DNA that is cleaved gets degraded, but for researchers, restriction enzymes can be used to stick pieces of DNA together using DNA ligase. Sticky ends are more useful for this because it allows for more control over how the DNA is stuck back together, as the sticky ends would want to pair up. PCR, which stands for polymerase chain reaction, is a process that allows us to amplify fragments of DNA. In other words, we can start with a very small amount of DNA, then use PCR to create additional copies of a specific region of DNA. PCR is used for many things, including genotyping, which is done to determine an individual's genotype, and detecting the presence of bacteria or viruses, which can be used to detect infectious diseases or identify contaminants in food. It is ultimately similar to replication and requires template DNA, a special DNA polymerase, usually referred to as Taq polymerase, nucleotides, and primers that provide a 3' end for the polymerase to work off. The primers are made to be complementary to sequences surrounding a region of interest that will be amplified. PCR has 3 steps. The first step is denaturation, which refers to the breaking of hydrogen bonds between the complementary base pairs and the separation of the 2 DNA strands. This generally occurs at high heat, around 95 degrees Celsius. The second step is annealing, which refers to the primers pairing with their complementary sequence on the DNA. This generally occurs around 50 to 60 degrees Celsius, with the temperature depending on characteristics of the primers. The third step is extension, when the polymerase essentially performs elongation from the primer. This occurs at 72 degrees Celsius, which is the optimal temperature for Taq polymerase to work at. Taq polymerase is special because it was isolated from organisms that live in hot springs, allowing it to remain intact even at high temperatures of denaturation. After the first round, we end up with double the number of DNA. The process then repeats with the new strands of DNA, and then the next. By the third round, there are fragments with a specific size. PCR usually runs for 30 to 40 cycles, with the amount of DNA doubling every cycle. The final product is mostly fragments of a specific size, determined by where the primers are, the size of the primers, and the amount of nucleotides between the primers. One of the things that can be done with the DNA after restriction enzyme use, or PCR, is gel electrophoresis, which is separation of molecules in a gel based on their characteristics. In this case, the DNA is separated by size. The gel used for DNA is agarose, and a current is used to move DNA and separate the different fragments. Due to its negative charge, DNA moves towards the positive end. Since the agarose gel forms small pores, smaller fragments are able to more easily pass through the pores, which allows them to travel more quickly. Generally, a ladder, which is a set of fragments with known sizes, is used to figure out the size of the bands. For example, in this ladder, the bottom band is 100 base pairs, going up to 200, 300, 400, 500, and eventually even 5,000 base pairs. The small fragments travel down faster. Based on the latter, I can say that this faint band here is probably around the size of 500 base pairs, and there are even more bands past the 1500 mark. DNA fingerprinting is a technique that combines the use of restriction enzymes with gel electrophoresis. It uses different sets of restriction enzymes to cut DNA into fragments, and these fragments will vary from individual to individual. Hence, they are essentially a unique fingerprint that can be used for probable identification of a person, such as for forensics or paternity testing. This image shows an example of how the DNA fragments would look in the mother, child, and 3 potential fathers. The child has DNA that matches up with the mother, and based on the remaining DNA sequences, the first person is most likely the father. Molecular cloning is the process of copying and amplifying recombinant DNA, which is DNA mixed from 2 different sources. This generally involves inserting a fragment of DNA, created by restriction enzymes, into a plasmid that was also cut open by the same restriction enzymes. The sticky ends created by the restriction enzymes will allow the ends of the fragment to pair up with the ends of the plasmid, and DNA ligase can be used to stick the fragment to the plasmid. However, the plasmid can also just stick back to itself, so it's important to be able to distinguish between plasmids that have the fragment inserted and plasmids that just return to normal. In this case, the plasmid has LAC-Z, the gene used to make beta-galactosidase to process lactose, and will turn blue if it has LAC-Z, and will not turn blue if the fragment was inserted in between the gene, which would prevent LAC-Z from being expressed. The plasmid can then be taken up by bacteria through transformation. The bacteria can then divide and make more copies of itself, along with a fragment of DNA inside the plasmid. Since only the bacteria that got the recombinant plasmid will be white, these bacteria can then be selected for further growth and propagation of the DNA fragment. CRISPR-Cas9 is a relatively new technique that has been developed for gene editing. Like restriction enzymes, it was originally discovered as a defense mechanism used by bacteria against viruses. CRISPR refers to specific palindromic sequences in the DNA, while Cas9 refers to an endonuclease that recognizes these sequences and makes double-stranded breaks. They are often compared to molecular scissors. In this example, we have the original DNA sequence in red and these blocks that are regions of interest. The Cas9 is taken to areas surrounding the region of interest and makes cuts, fragmenting the DNA. In some cases, a template can be added so that the repair mechanisms use the template for repair, resulting in recombination and edited DNA. CRISPR-Cas9 requires guide RNA, abbreviated gRNA, that essentially serves as a guide and takes the Cas9 to a specific sequence of DNA in the genome. The Cas9 then makes a cut in the DNA, and the cut DNA can be repaired by DNA repair enzymes, and sometimes a template that ensures that the DNA is edited in a specific way. Biotechnology has been very beneficial for medicine. For example, the development of cloning allowed for mass production of insulin to treat diabetes, as well as other peptide hormones. It has also allowed for production of vaccines and antibodies to combat diseases. We can also now perform genetic testing to find out if we have genes that put us at risk for certain diseases, such as breast cancer. With CRISPR-Cas9, gene therapy is now a possibility, and there are many single gene mutations that can potentially be cured one day using this technology. Biotechnology has also been helpful for agriculture. We can now screen the DNA of plants to find genes that encode resistance to drought or to infections, or for better growth. Plants that have these characteristics can then be chosen for breeding, speeding up the process of breeding plants with beneficial characteristics. While more controversial, biotechnology can also be used to create genetically modified plants and other organisms for food, adding in genes such as resistance to pesticides or pathogens, or in the case of golden rice, making the rice produce carotene. Carotene is naturally made in the non-edible parts of rice, and golden rice was modified so that it would also be made in the edible parts to help solve vitamin A deficiency. This brings us to bioethics, the study of ethical, social, and legal issues that arise from biomedical and biotechnological research. As we continue learning about biology and its applications, we want to make sure that the tools we are using are ethical and safe. That's why there are many rules and regulations in place for every aspect of biotechnology, and these regulations will continue to develop as we learn more information. There are many questions, concerns, and considerations that remain. For example, the CRISPR-Cas9 system can be used to correct single gene mutations to prevent genetic disease, but this should only be targeted to somatic cells, not germline cells that can be passed on to descendants. It is currently illegal to use CRISPR-Cas9 to genetically modify embryos, as that would affect all cells in the body, including the germline cells that can pass on the genetic modifications to future children. That's why it was incredibly controversial when a scientist revealed in 2018 that he had used CRISPR in an attempt to make 2 babies resistant to HIV. Furthermore, there is a distinction between using CRISPR-Cas9 to correct mutations that cause disease and using it to obtain specific traits. As more ethical concerns arise, it is important for us to discuss with one another, share our thoughts, and listen. In this video, we learned that restriction enzymes cut at specific palindromic sequences, PCR is used to amplify specific sequences of DNA, and gel electrophoresis is used to separate fragments of DNA. We learned that DNA fingerprinting can be used to help identify suspects or be used for paternity testing, and molecular cloning can be used to create recombinant DNA. CRISPR-Cas9 is a technique that has been developed for gene editing and involves using a guide RNA to bring the Cas9 to a specific region of DNA, where it can make a cut that can be repaired with an edited template. Biotechnology has been beneficial for medical treatments and producing better crops, but there are many ethical concerns that remain that are important to discuss.